but it's a lot of mammals right around that Eocene-Oligocene boundary that are here in North America. I just had the fun of getting to work on them in a different way. So just to give you an idea of some other things that were going on around that time, or just before that. These are fossils you may be aware of in that time. Jacob, could you please? I don't know why this isn't working better. Sorry. So the fossils I'm working on have actually been known to scientists and to museums for a very long time, actually for over a century. Because while in the last century and even a little bit before that, there were people very excited about going to Egypt to look for mummies, there were also people who looked around and realized that there were these big, amazing animals that they had never seen the fossils of before in Egypt, in Africa, that aren't anywhere else in the world. And so there were these big, charismatic animals with weird horns on their faces that they didn't know what they were. That means it's time to pick up a field expedition and get going and go find some more of them. And that meant that everybody needed one. So the American Museum of Natural History actually did a dig in the early 1900s, and a couple of European museums before them, looking for these big animals that weren't anywhere else in the world. And this guy is Arsinotherium, and we will come back to him because he's really, really special to this place. But just so I can orient you in time, at that time, people were kind of more or less trophy hunting. Oh, there's big, cool skulls. Let's go get one to show off in our museum. And science has come a long way since then. So now we have the fossils. A lot of them have been named. We can start to tell stories with them. And it turns out that these fossils are from a really important time in Earth's history. Like I said, about 34 million years ago. And this was a time when it went from being really, really hot to being very cold. And that change happened very quickly. Jacob, would you please click? Okay, here, if you'll hand me my mouse, I can actually.
I got another clicker for you to try.
Okay, we will use another clicker. Sorry about that. So it went from being very hot to very cold very quickly. And when I say very quickly, in geologic time, I know we use that term differently depending on what scale you're looking at. We're talking on the order of centuries to thousands of years, which when you're talking about millions of years is very fast. But because it started to get colder, that also meant that we started to have ice at least the South Pole, potentially also the North Pole, for the first time. Once you have ice on the poles, everything else starts to change. It changes the currents that are going around the poles. And because you change water currents and air currents, that also changes all of your weather patterns. This is actually part of the reason we talk about this now, because this is, of course, a concern today. As the world heats up, we're starting to lose ice, and it's not just, oh no, we're losing ice, not the polar bears. The problem is that that affects everything else. It's going to affect weather patterns. It's going to affect probably things that we can't even begin to imagine yet because our models can only take us so far. But the most important part to this story that I have is that it's not just changing temperature. It's also changing how arid or how wet a place is. And those changes aren't going to happen the same in every place. So there's some gradient with latitude where the poles are probably going to be a little more affected than the equator. And then there's differences if you're coastal or if you're not coastal. And there's a lot of local factors that go into just how much you're affected by what's going on in the world, the rest of the world around you. So while this is a very fast global change, we might not necessarily know what was going on in an individual place. Part of what happened was in the Eocene going into the Oligocene was because water was getting sucked up into ice. That also meant that the sea level was going down. And because the sea level was higher at that time, Africa had actually been isolated from other continents for a very long time. And this isn't just important to climate patterns, this is also going to end up being important to what animals were there, but we'll come back to that part of the story. So as I said, your temperature changes, your ice changes, this also changes what your coastline looks like. It's also going to impact aridity or how much rain most places get. And in most of the world during the Eocene-Oligocene boundary is what we call it. We went from having a hot, humid, wet, rainy climate in most places to most places getting very, very dry. And when you lose water or lose rain, that also is going to change the plants. Dr. Mickle, I promised. This is also going to change your plant community. In general, if we were in an area that was very forested, those places that were forested tended to lose forest. It probably didn't get quite as dry as a desert like my picture. That might be an exaggeration, but it did start to look a little bit more like a prairie, other than the fact that we didn't have grass yet. But think a very open habitat. And if you change the plants, you also change the animals. So instead of animals that were climbing in trees or eating fruit or eating leaves, you had more animals that were better adapted to that open habitat and were more adapted to eating, again, not grass yet, but things that are growing on the ground instead of things that are growing in trees. We also, another important part of the story is we almost lost primates at this time. Primates used to live over around most of the planet. But in North America, Asia, Europe, South America, I believe, most of the primates went extinct. No primates, no us in the future. That was close. But it turns out they actually survived in Africa. And for some reason, the reasons we're starting to understand, something was sort of protecting them from this. And there's a couple of reasons that we're going to get into what might have saved them for this. And this is part of why people are so interested in this. If you look, these are places that we kind of know what was happening during the Eocene-Oligocene boundary, or where we only have part of the fossil record either before or after it. So not everywhere, and basically all I wanted you to get out of this is that it was different everywhere. It didn't always go from hot to cold and from wet to dry. It depended on where you were, and we're starting to understand that as we find more fossil sites that have a span of time that can tell us that whole story, because we don't have that from everywhere. And Africa is not an easy place to study, even though we have been excavating in some parts of the continent there for a century. There are challenges to getting there and to excavating, and be they political, be they just geographic. There's a lot of reasons that Africa is understudied. And also, a lot of the fossils are in the desert. That is not a very accessible place to go on an excavation. So we actually don't know a lot about what was happening in Africa, really in the past at all, but especially during this particular time. It turns out, though, that Egypt is one fabulous place, one amazing place where a lot of fossils, especially from the Eocene-Oligocene boundary, were preserved. Where really on most of the rest of the continent, we haven't found anything yet. We have a lot of material from here. It's an amazing record, which is great because that's going to help us fill in part of the story. Now, because Africa was isolated from kind of the rest of the world at the time, that means that animals, unless they were swimming across this, for the most part, the animals that were in Africa were the animals in Africa, and the animals that were in Europe, Asia, everywhere else were the animals in everywhere else. So where in some parts of the world the Eocene-Oligocene boundary happened, new animals that were better adapted to those environments kind of took over. And if they took over in one place, they tended to spread to other places. Africa was really isolated from competition that might have created. So animals like at this time, you got more ungulates, more big grazers, and a lot of new carnivores were evolving and taking over the rest of the world at this time. Africa didn't really have those guys come in, where instead Afrotheres, or animals that originated in Africa, like actually the hyraxes, which are still around today and today look like this, basically a groundhog with tusks that's related to elephants, the ancestors of elephants, and then that weird Arsinotherium that we don't really have. We don't have any relatives around of anymore, other than the fact that it is somewhat related to elephants. Those guys they persisted, and they were doing just fine on this continent. They didn't have anything competing with them other than themselves. That also meant that the carnivorous mammals that were in Africa weren't being outcompeted by more modern carnivores. And the primates, for whatever reason, survived in Africa a little bit longer than they did everywhere else. I say a little bit longer, I actually mean survived until today. And just to give you an idea of what was going on, part of why this place is so fossiliferous is because of the environment it was in. And part of it's just because we know it because we've been looking for things in Egypt for a very long time, been interested in that area for a while. But part of what helps that is it was a coastal site. There was a lot of deposition of sediments. Things were getting buried as soon as they died and are in a place where they preserved. So about 37.4 million years ago, those are the first terrestrial land fossils that we see in this environment. And it was probably a coastal area. Actually, most of that formation is this marine nearshore environment. It's known for whales, it's known for early whales, seen some shark material from it. It's a very fascinating site, and there's a whole different group of people that study the marine mammals out of that site. But there's this one little blip in it that's kind of a terrestrial island. I like to think of it as kind of our Outer Banks in North Carolina, basically a sandbar where a few terrestrial animals got buried. A little later in time, about 34.6 million years ago, the ocean had started to recede a little bit. And this happened in pulses. It didn't happen all at once. And it left behind kind of a lake environment that things fell in, they died, there they were, they got buried, and we would find them 34.6 million years later. And that environment has some petrified wood around it. There's some evidence of some mangroves around, which are something you'll see in an area that still has the salt stress of being near the ocean. So it's probably not entirely a freshwater environment. It's probably getting some saltwater flooding once in a while, but it's a little quieter. It's not the coastal, it's not the same coast that it was before. And after that, we have later localities that still have the mangroves around. And there's a lot of argument about these. We're not really sure whether it kept being the same kind of mangrove forest that it was before. If it became much more open, which is what we would think based on the Eocene-Oligocene boundary, or if it kind of became a denser forest and less of a mangrove forest and more of a true forest where the primates were eating fruit. And that argument is kind of entirely up to now, has entirely been based on, we have primates. The primates that are here after the Eocene-Oligocene boundary are more arboreal and look like they have teeth that would be better for eating fruit. Versus people who are saying, no, that's an Eocene-Oligocene boundary site. In the Oligocene, it should have gotten more open. We lost the forest instead of it becoming more forested. And then there's people who look at the community as a whole, at the animals as a whole, and see, well, the animals change. Which animals are there change? But the things that we think they were doing based on their teeth and based on their size, the body sizes represented don't really change, the diets don't really change, the environment was probably pretty much staying the same through time. So this has been an argument, and it's been entirely based on the morphology of the fossils, which is great, but we can do better. Or we can add to that, I guess I should say. And just to give you an idea of the animals that are there, I already mentioned some of them because they were special to Africa, but the thing is, when you get into the Fayum, which is the site I'm working on in Egypt, it's most of what we know about the African fossil record from this time. So these are the animals that are known from this site. It's known for hyenodonts, which are carnivorous, but they are not carnivora, which is a pain in the butt. They were eating meat, but they are not related to dogs and cats. They are their own thing that are entirely extinct as far as we know. And there were also this really scarce thing called a Ptolemaid, which is a carnivore. It is another carnivorous mammal. It's small. We don't have a lot of material from it. They're very rare. I have not been able to work on these guys because they are so small and they are so scarce, but we're working towards that. You have, as I mentioned, the ancestors of elephants, and there's 4 of them at this site. There's Moratherium, which looks very, very hippo-ish, looks like it should have been semi-aquatic, has short legs, probably a short trunk. very chubby, very round. That's this guy up in the corner. You have Baratherium, which is probably similar. It's another early elephant. And then you have Paleomastodon and Phiomia, which kind of gets grouped in with Paleomastodon sometimes, both of which could have been partly semi-aquatic, but they are also found in more terrestrial environments. So where Moratherium and Baratherium are almost always found in gross, swampy, there was definitely water here all the time environments. The other two are generally, they can be found there, they can be found in forested environments. There were two other Proboscidean ancestors floating around the landscape at this time that are only found in more terrestrial environments and never found in those swampier areas. They are not here. So already that tells us a little bit about this site. You should know as we go forward that anything that's related, any of the Aphrotheres, there is a chance that they came from something semi-aquatic. This is part of their history and part of their story. And there's always a question when we're looking at these guys of just how semi-aquatic were they. So there's also the Hyraxes, which again are the closest modern relatives that elephants have, which is really funny when you look at this guy, but not so crazy sounding when you look at its ancestor Titanohyrax. And while today they're about the size of a cat, you haven't seen them because they only live in Africa. In the past, they could be anywhere from the size of a cat or even a little smaller to the size of a rhinoceros. They varied a lot in size. They varied a lot in their tooth shape. They varied some in their postcrania and their body shape, but the fossils are kind of limited, so there's only so much we can say about that. This particular one that's in the picture, I should have had a scale, but it's probably about this deep of its mandible. They are large, and then there's also the small ones running around at the same time. And we really don't know that much more about them, except that in the past they were so much more diverse. And then we have the Anthracotheres, which are one of my members of the story that are actually not related to elephants. We finally get something a little bit different here. But the Anthracotheres are probably the ancestors of hippos. And where we were living in Cincinnati, some people know her, some people don't. Fiona is a very famous modern hippo, so she has to make her way into this. But hippos even, it's important to keep in mind, lived in Egypt until very recently, and if it wasn't for human impact, the human impact of building a dam would still be there. So it's kind of interesting that they continued to be a part of the story for a while. And again, with Anthracotheres, there's this continuous argument in paleontology over whether they were semi-aquatic or not, and when exactly in their history they started acting like hippos. And they kind of look like they should be acting like hippos. Again, they have a low body, short legs, but it's really hard to answer to what degree that was happening. And another thing to keep in mind, and we'll get into this later, is their teeth are actually really similar to some of the larger hyraxes, to the point that if you're working in these collections, there are a lot of places that the word hyrax is crossed out and Anthracothere is written next to it, or vice versa, because things have been identified as one in the field and then they get back and go, oh no, that's the other one. They're very similar teeth and there's overlap in size. These guys didn't get that small, but the hyraxes got that big. And then, like I mentioned a few times now, you've got Arsinotherium, which is this large rhino-like thing that is completely extinct now. And Arsinotherium, we don't really know that much about it, other than it looks like a rhino and was probably a browser or herbivore that wasn't grazing. That's probably close enough. So thinking of it as basically a rhino is probably close enough. But again, it's a large ancient mammal, and people like to argue over whether it was semi-aquatic or not. This is a constant conversation that we have. And as I introduced you to before, this is also a place that's known for petrified wood and known for whales. And of course, it's very famous for its primates because it's one of the only places that we have a record of them. But it's also one of the only places that they survived through this crisis that's happening. And this is where I start to come into the story, because in several decades ago, we switched from people being really interested in the large animals, which we still are. We still want to know what the large animals were doing. to places like Duke being very curious about the primates. Curious about the primates, these are the ecosystems you study. So Duke actually has a really phenomenal collection of fossils from Thiam. They're continuing to do excavations, and they are actually starting to work very closely with paleontologists that are in Egypt. We finally have some colleagues in Egypt, which is amazing. To keep those excavations going, to make sure that that they're being studied by both people here and there, and to make sure that education is happening in both places as well. So there's an amazing collection in our own backyard of these fossils. And as I said before, I get so excited because we can describe these fossils and think about, oh, well, that's the shape of an animal that would have this habitat. But it's a different question to ask what they were actually doing. And we're coming into a world where we can actually start to ask those questions with new methods that are ever getting better and constantly, because they're constantly improving, we're constantly able to ask more of those questions and ask them better and ask them without completely destroying fossils. That was a big jump. And so this is exciting to me. So I wanted to introduce you guys also to these awesome methods that I'm using. So I do stable isotope ecology. You've had a couple people talk about them in here, that method in here before. And essentially, the questions I'm asking aren't what is this animal adapted to do long-term to survive? If everything goes wrong, what is the worst case scenario of what it could survive eating? I'm asking questions more about what was the population in a given time and place actually eating based on what was available to it. So I'm going to embarrass my husband because he actually helps me as a really good example of this. Human teeth, all of our teeth more or less look the same. There's some variation, but in a few hundred years, when future archaeologists, paleontologists have changed what they're eating and forgot what we were eating now, they would look at the skull of any one of us and say, oh, those are the teeth of an omnivore. Probably. You've got teeth that can cut and slice, but you've also got teeth that can grind. So you can and should be eating both plants and meat, and everything else. So this is my friend Julia over on the left. And she is a fellow paleontologist, does a lot of really amazing work. Julia is a vegetarian. She chooses not to eat meat. So there's a difference between what somebody would see looking at her skull and what somebody who knows her or uses my methods would know by taking a sample of her teeth and running the chemistry on it. So running that chemistry, I could look and say, oh, she probably wasn't eating meat. She's too low of a trophic level. And we'll get into how I can do that. My husband, on the other hand, really does not like vegetables for the most part. He is her polar opposite. And if in the future somebody takes a sample of his teeth, even though his teeth basically look the same as hers, the chemistry of his teeth is going to look dramatically different, and they're going to be able to say, oh, wow, that's somebody that was eating a lot of meat and not a lot of veggies. Now, this also becomes important not just because of what you were actually eating versus what you are adapted to be able to eat, because I would hope, Jacob, if you were starving, you would pick up some vegetables. This isn't just about a worst case scenario, but it's also about what happens when other animals come into the picture. So if our buddy Tim here came in and created competition with Jacob, because Tim also does not like his vegetables, but he sure loves meat, and he will fight you for whatever is on your plate. There's now competition created, and if one of them eats all the meat in the house, the other one, not going to say who wins out in that battle, has to find something else to eat instead. So there's this there's what you're able to eat altogether, and there's what you're actually doing, what niche you're actually filling, based on what is really available to you and based on what the other animals around you are doing. And if you're in a place where climate is changing very quickly, and the food sources are changing, the habitat's changing, what you're actually eating might change even if the shape of your teeth doesn't change, even if you don't go extinct and something else with a different shaped teeth shows up to eat different food instead. So this is where we get into fun graphs and pictures. And if you are either in a place in school where you haven't been introduced to chemistry yet, or if you just see a graph and glaze over and you just about walked out of the room and turned your computer off, I invite you to look over here in the yellow square. Place for you. because this is where I am going to show you what's happening as best I can with illustrations and translate it into something a little kinder than chemistry and math. If you are actually interested in the chemistry and the math and the graphs, I have that for you here too. So isotope ecology and that fancy chemistry that I run boils down to you are what you eat, what you drink, where you live, all of that. And it's a lot more complicated than this. We're just going to keep it simple. But essentially, the carbon in the food that you eat changes based on where the plants that you're eating or the plants that what you're eating ate, where they grew. And so if they grew in a really dense forest or in a wet environment, they're going to have lower values and be down on this left side of the graph. And if they're in a dry environment or somewhere that's water stressed, think salt, they're going to have a higher value and be over on the right side of the graph. And then oxygen isotopes are more or less tied to what you're drinking and how you sweat and what your body physiology is and your body size. And let's just stick to it's tied to your body water. It's a lot simpler. We joke that oxygen is a nightmare because if you sneeze on it wrong, it changes. But essentially, it's tied to water. So if you're something living in a really damp cold environment where a lot of evaporation is happening slower, that damp, cold, wet environment is going to be lower on the graph. And if you're somewhere dry, you're not getting a lot of water, you're, for whatever reason, it looks like you're water stressed, you're going to have a higher number. You're going to be higher up on the graph. So just to give you an idea of where some different ecosystems and different diets could fall on this graph, if you're something living in a marine environment, you're going to have kind of low oxygen, but higher carbon because of that salt. And you're going to have a very kind of narrow variation if you're swimming. Versus if you're a terrestrial animal, you might have a friend that whose values are up here, your values might be way down here, and it averages out to having a very, very wide variation in values. Where aquatic animals, it kind of squishes down and they're all doing something very similar. If you're living in freshwater, same story, except that you're going to be a little bit lower and on this side of the graph that's a little less water stressed. And if you're brackish is between fresh and salt water, you're somewhere in between. And then terrestrial animals, like I said, much more variable values. If you're in a more forested area, you're going to have isotopes that look like you're somewhere wetter than if you're living somewhere dry or open. And then there's, if you're eating grass, which is not relevant to this story, you'd be even further up and off this graph. But we're not going to worry about them because we don't have that much grass yet. And then within those environments, we can get even more specific to a point. So if you're living on the forest floor, you're going to have even more wet looking isotopes than if you're living in the forest edge or in the canopy of a forest, of where it is a little bit drier, it's not as dense, there's more sunlight, there's more wind. Really, this is way more complicated than it should be, and yet we enjoy it so much. Now, the problem is with these isotopes, we only have a two-dimensional space and a lot of different things that can happen. So there's overlap, and sometimes it's a little hard to interpret whether something is, you know, say in a forest canopy or in a forest edge, or it was actually in a dry environment and you just need to kind of shift graph a little bit. Or if it was in freshwater or in a dense forest. Sometimes these are hard questions to answer. So it actually works best if we use multiple other methods. And this is where an undergraduate that I'm working with at Duke comes into the picture. My mentee, Orion, has been doing a lot of work photographing as many Hyrax mandibles as he can. I think he just about got sick of it, and yeah, he's doing a great job. And he's helping me look at how fast the teeth of these Hyraxes warp. So basically, the faster the wear on the teeth, the more abrasive the animal's diet was, which tends to be something we associate with a more open habitat versus a more forested habitat. So where I'm dealing with that overlap in some places in the isotope record, he's helping me back up our interpretations with the wear of the teeth. And so he looks at multiple individuals from one species from one time and compares how extreme the wear on the individual's teeth from that species is compared to how extreme the wear is on another species, in individuals of another species. So he does that by helping me measure the height and the angles of those teeth. And basically, as I said, the faster the wear, the more abrasive the diet, the more likely you are to have been eating grass. There's some arguments about why exactly this is happening. We don't need to get into that, but it's a good proxy for diet, regardless of what's causing it. Now, this is where that grant came in handy, and I wanted you guys to get to hear like exactly where that went. So we had the amazing opportunity to go to Germany and to Egypt last summer. Germany invited me because they're actually the ones running my samples. Where normally you have to pay a lot of money for isotope samples to be run, Germany covers the cost of that research because they're very excited about science being done. They're great. So they, working with the Max Planck Institute for Chemistry, they were willing to run my samples because they're testing a new method. So I got invited to come learn how that method works. And basically what they've done is they've tinkered with how we normally do this chemistry to the point that they can work with a much smaller sample than has historically been able to be used. And because they're using a smaller sample, I can sample those small hyraxes. And we're hoping eventually I'll be able to sample the primates and even the hyaenodonts. Stay tuned for that in the future. Right now we're working on the large mammals that are a little more abundant and a little less protected by Duke. And you know, it turns out if you get to Europe, getting to Egypt is a whole lot easier. So we also had the opportunity to go see the field site and actually sample some fossils that are in the care of Dr. Salam and his students, the paleontologist that's in Egypt right now. Because it turns out that going and sampling fossils in Egypt is a lot easier than it is to ship fossils across the ocean to sample here and then ship them back. This was much easier and made for a much more interesting trip. So that also gave us the opportunity to see the field site and go to some really incredible places. We got to sample the fossils, and it also meant that I got to train one of his students in what we're doing. So where they're mostly studying morphology, she actually studies the rodents and the hyaenodonts at the site. They don't have an isotope person right now, so they got to learn about how I sample and what's being done on my end to kind of back up what they're learning. So I wanted to show you guys what my results were. And as I said, if this scares you, I have a box for you. So anyway, in that first site, that 37.4 million year old site, we've got basically two animals we've been able to sample. One of them is the proboscideans. It's Morphtherium. It's that one that is probably definitely semi-aquatic, and we know that from the morphology. And the isotopes back that up. It's down in this low space with low carbon, relatively low oxygen, but it's a little bit higher than I would have expected for carbon. And we're taking that to mean, yes, this was definitely a coastal environment. We knew that from the geology, but it's swimming in that brackish or salty water that was available. It also has a higher variation in oxygen than I would have expected for an aquatic animal, but the morphology really looks like it should be aquatic. So there's actually a good chance that this guy was acting a little bit like a hippo. If you know anything about hippos, they never actually look aquatic when it comes to isotopes. They're very annoying because we can look at them and go, no, we know you're spending your time in the water, but your isotopes don't look like it. Morphtherium looks like a hippo. And by that, I mean, it has isotopes that look like it was probably maybe spending its time in the water, but it doesn't necessarily look like that because it was probably getting out of the water and going and eating something else and then coming back and going for a swim after it eats. So it's not necessarily eating in the water where it's spending most of its time. As I said, hippos like to break the rules. And then we have Dimetherium, which is a very small hyrax, very early hyrax, and it's the only one from this site. This guy also likes to break the rules. So this hyrax has higher carbon and oxygen values than I would have expected for anything semi-aquatic, but it's narrow. It's a low amount of variation for an animal that's living on land. There's no animal that lives on land that I've found that has this low of variation. So that means one of two things. Either it's eating a really specific diet or it's breaking the rules and is behaving in a semi-aquatic way, perhaps in a marine environment, which would explain that higher carbon and even the higher oxygen. So again, semi-aquatic animal tends to squish the values down to have very low variation. And that meant that my advisor and my committee had a lot of questions because this hyrax looks like hyrax. It doesn't look like it should be swimming. But we get into all early amphitheaters have this one gene that's basically a diving gene, a diving protein. That means they're probably coming from something semi-aquatic. Hyraxes today, we're kind of limited to hyraxes that live in the forest and the desert. Don't really have the opportunity or need to swim. This guy was living in a coastal environment. So my advisor asked, well, what could he have been eating that had that low of variation? Good. Okay. What could have been eating that was that specific? Turns out his isotopes look an awful lot like animals that are eating seagrass. And there was seagrass in this environment. She goes, okay, you gave me a food that was there that it could have been eating. I'm okay with this. But is there anything today that looks like it's semi-aquatic or doesn't look like it's a swimming animal that dives and eats seagrass? Because hyraxes really don't look like they should be diving and eating seagrass. Well, y'all heard about moose? They sure don't look like they should be swimming. But they have been known to dive deep and graze off of deep plants. And we've known that they've done this in lakes for a long time. But actually, as we get into their isotopes, even in the modern day, turns out they're doing that a lot more often than we thought they were. And the chemistry of their teeth supports that. And she goes, okay, I guess I'll allow for that. You answered my questions. And so I said, now I have a tongue twister for outreach, and it makes me so happy. So I give you, and in case you don't know, seagrass is in a group of plants called macrophytes. So now I have my mini marine moose, sadly munching many Mediterranean macrophytes. If you can say it more than once fast, I will be very impressed. So anyway, we've got hyraxes and Moritherium, both of them behaving in a semi-aquatic way, but doing it a little bit differently in this coastal environment. Now, you go a little bit younger, and this is still before that dramatic climate change event, but it's as the sea started to recede. And now we just have Anthracotheres and more hyraxes, but still hyraxes. Moritherium, I don't have available to sample from this site. I don't think it's actually been found at this locality. Not sure where he went. This is that one that looks like a lake. But we have Anthracotheres and hyraxes, which, if you'll remember, some of the larger hyraxes look an awful lot like the Anthracotheres. And they're overlapping in space. So they're probably eating something pretty similar. And they're in this higher space, there's more variability. They're probably mostly terrestrial animals. And they're kind of in this space that I would expect them to be in if they were in a more open environment. I'm sure there's trees somewhere, but they have a lot of different foods available to them. And it's important to keep in mind at this time, we go from having one hyrax to a wide variety of hyraxes, including very large ones, including very small ones. And they're not all doing the same thing. So on my graph, if I were to break it down, the large hyraxes are actually in a higher, more grazing space, let's call it, than the smaller hyraxes. And the smaller hyraxes are overlapping in space with the Anthracotheres more than the large hyraxes were. So remember that whole thing about when this can reveal competition? These guys, the large hyraxes that have similar teeth and similar size to the Anthracotheres, aren't filling the exact same space as the Anthracotheres. So they're probably eating something slightly different and using a slightly different habitat. I can't tell you exactly what that is. Those values are really close to each other still, but there is some separation happening. So that's really cool because that tells us there are these things that have similar body size and similar shapes are dealing with that, are avoiding competition by living and eating in a different place. As I said, their teeth, really similar, and body sizes are very, very similar. This is where my friend Orion, my mentee and friend Orion comes into the story a little bit. Just to give you a short version of what he's been working on, because I don't want to completely steal his thunder where he's presenting at a conference later this year. Remember, if you'll think back to where I said faster wear means an animal is eating different things. the hyraxes have different rates of wear in this locality, where it looks like our larger hyraxes are spending more time grazing than our smaller hyraxes. And this isn't the best example I have, and I apologize for that. But pretty much it looks like the larger hyraxes are essentially wearing their teeth out earlier than some of the small ones. So they're in that open habitat grazing more than the smaller hyraxes are. So it's really nice to have that method, that wear, backing up our isotope work. That's exactly what we want to see. Now you would expect at the Eocene Oligocene boundary the world to get drier, because that's what happened in most places. If it got drier, the carbon and oxygen values should go up, right? Science always goes exactly as expected. Wrong direction, but that's okay, because this was the alternate hypothesis. This was the other possibility based on what people were seeing in the primates. And so it actually looks like instead of more open habitat becoming available, more closed habitats started to become available, more forested habitats started to become available, excuse me. So my hyraxes and brachytheres, many of which were there before, are using this more forested space now and aren't in that open habitat. But it doesn't look like that habitat was completely gone, because I've got my other two elephants, my other two proboscideans. And in this case, it is Paleomastodon, and I think I have Baratherium in there. It's basically my other two proboscideans are spending time in that more open environment, exactly where the hyraxes were before. Again, this is a method that can help reveal when competition is happening. Apparently, these new proboscideans showed up on the landscape, started using that open habitat, and the Anthracotheres and hyraxes realized, well, there's more food available in the forest, there's different foods available in the forest. We can eat the food in the forest. We don't have to use the same habitat and the same foods that these elephants have shown up using now. At least that's my hypothesis on this, because it's really the main reason I can think of that these guys would shift to using that forested habitat. They don't really change that much morphologically, and they don't really change which Anthracotheres and which hyraxes are there. And this is immediately after that change happens, that climate change happens. So now we've got elephants living in a more open environment that's probably still available, and Anthracotheres and hyraxes living in that more forested environment. Now the hyraxes are kind of in between, and if I break it down, I actually have some hyraxes kind of sneaking into elephant space up there, let's call it. So they're probably spending time in both of those habitats, and that's okay. The smaller ones probably aren't competing are not directly competing more than likely with those elephants, but they're able to use both of those habitats and probably using the forest a little bit more than they are using the space. Anthracotheres, they just wanted nothing to do with it. The thing to keep in mind, though, is there might have been some competition happening, because at this point, the large hyraxes are completely gone. They're not extinct because they come back later, but they're not living at the Phyam for their fossils to preserve. And this is a locality that is known for large animals. It's known for the elephants. It's known for the Anthracotheres. Why wouldn't it preserve the large hyraxes? So we assume that it's probably a safe bet that the hyraxes the large hyraxes just weren't on the landscape for some reason. I'm willing to blame competition for that for right now. And then we keep moving forward in time, and we keep seeing the hyraxes, the Anthracotheres, and now I have our Arsenitherium using that lower forest space. And the hyraxes shift again. Our Arsenitherium shows up. It's using a little bit of kind of that edge space that I was referring to, that in between forest and open environment. And the hyraxes go, we want nothing to do with that. We're just going to go use the forest and shift a little bit deeper into that wet space. But at this point, and in the next locality, the larger hyraxes come back. Like I said, they weren't extinct. They just didn't want anything to do with the elephants. And so this is a little bit later. This is 29.8 million years ago. It's moving forward in time, but still you see forest, you see everything down in that bottom corner. Still have Anthracotheres, hyraxes, and the big rhino-looking Arsenitherium. You still have hyraxes and anthracotheres using that much denser forest space versus the Arsinotherium using kind of this edge. And like I said, the big hyraxes came back. Now, what's really cool about this, I didn't really get into all of the different diet things we can tell, is that our larger hyraxes and smaller hyraxes, because they're splitting like that, the smaller hyraxes are in high carbon, low oxygen space, and the large hyraxes are for that. Well, normally, one way we can look at that is higher carbon, lower oxygen can reflect an animal eating more fruit, and higher oxygen, lower carbon. I didn't need another tongue twister. I've already got it and built into the science. Higher oxygen, lower carbon can be an animal that's eating more leaves. So we don't just have some habitat splitting here, we also have some diet splitting. And there's a good chance that these smaller hyraxes are eating fruit. And they actually, several of them have a tooth shape that we associate with a fruit eater, while the larger hyraxes have more of a tooth shape that we associate with plant eater, or leaf eater. There is one large hyrax down in this quarter, and it's a hyrax that's bunodont that has teeth shaped like ours and like most modern primates that, again, we associate with an animal eating fruit. So that kind of backs this up, that these guys are probably being a little more picky in what they're eating, and the other ones are probably spending more time eating leaves and foliage. More to come on that as we get a better sample, because our sample size in this area is not great, but it's getting better. The problem is with this is that I'm missing proboscideans from later in time. So I don't know if the open environment was just gone by the time we get into everything spending its time down here, or if I'm just not sampling the animals living in that space. And there's a few things we can do to address that. So in addition to me finding proboscideans I can sample from those times, which I don't know that they're available. I keep looking, haven't found one from those localities that I can sample. The other things we can do is we can move forward towards sampling the hyaenodons and the primates. Hyaenodons were carnivorous. They have a habit of kind of averaging together everything they're eating. And so if they're eating things from different habitats, they're going to kind of show us an average of what the landscape looked like. Or if they're being picky in what they're eating, they might pick up a habitat that I wasn't able to sample from my hyraxes. So if they're eating something that's in that more open habitat and not the hyraxes, then I know that open habitat was still there. And then of course, the primates will kind of help us answer questions about what that forest looked like. They might back up that fruit versus foliage question that we have with the hyraxes. There's a lot more to answer. And also we're just really curious about primates because people want to know about primates. So we're really excited about the results we have. And we're really I'm really crossing my fingers that I can move forward and hopefully sample the hyaenodons and primates in the future. I do want to draw people's attention to this because a cornerstone of this club is outreach. Duke has scanned every specimen that I've sampled and more and uploaded it to Morphosource, which the last time I checked, it was offline for a little while, is back. And so these are great 3D renderings of the fossils that I'm working with and that other museums have. And I believe it's actually possible to 3D print from them. So if you ever want to see them, it is an online resource that's available to you. So with that, I have to thank UC, Max Planck Institute, and you and 2 other groups that provided funding for this research. And if anyone has questions, I would be very excited to answer them, or try to answer them. Yes.
Okay, so you mentioned Ptolemy. That's wait, you said it was a carnivore, right?
Ptolemy is it's really the only carnivore that we have from that site.
I thought it was also like I thought it was also an aphrothere. I remember like group called known as Ptolemaids that were like a group of aphrotheres from carnivorous aphrotheres that like lasted until like the myosin that I remember like other like genus like Cleopatrodon and Kelbo. Kelbo was like the very last of them that lived in the middle Miocene of Kenya, I think.
There's so little material from them that I think there's even some argument about exactly where to put them. And we could drop them into Aphrotheria, could drop them into carnivore. There's not a lot of material. When the last I saw, I was pretty sure they were in carnivore, but There's argument about it. There's even argument about the hyenodonts, and exactly where they fit into the story of mammal evolution.
Yeah, I've heard that the main group that they've been put in was known as Creodons, and also like the Oxyenids went into. It's
Yes.
been argued as paraphyletic, or wait, it's called
Paraphyletic.
okay,
You're
paraphyletic.
right.
I heard that little clade might be disbanded.
Yep.
And I could have sworn I heard somewhere that hyenodonts that the proposed closest relative for hyenodonts is pangolins, but I can't. But take that with a grain of salt. I don't. I think we just don't.
We really don't know what to do with the Creodons.
They're-
They are a very strange group.
Yeah, they're enigmas.
Yes. Even aphrotheres, we really don't know what to do with. You get to that higher level of how all mammals are related, and it's just a mess. I like my ecology.
I didn't hear you mention where you got your samples, to measure max length.
So they're all off of teeth that were either from Duke or in Egypt.
Teeth.
Yes, so oh wow, sorry. I did, I'm sorry. So what happened? Let's go back to that picture actually. Maybe, eventually. No, there it is. Oh, so what happens is, imagine going to the dentist and getting your actually science. That's the moral story. There we go. All right, so imagine going to the dentist and getting your teeth cleaned. Instead, they have a little drill that sounds really, really scary, and they are, you hope, just kind of clean black and gross stuff off of your teeth. I'm actually giving these teeth cavities. Not quite that bad, but I'm taking a little tiny drill, actually with dental picks, and removing a little bit of powdered enamel from each tooth. In the past, doing that from teeth, and some people will do it from bones, basically required doing away with the entire fossil. Part of what Max Planck is allowing us to do is to take so little material that I went to ask of my committee member at Duke. Y'all know Matt Forth. Some people in here know Matt Forth. Asked his permission to work on smaller animals and handed him one of the elephant teeth I had sampled, and he goes, "Where is it? What did you take any off this one?" So it's so small that it's negligible. Just about. It leaves a scratch. You could see it under a microscope, but we're removing a very small amount of material, and that powder gets dissolved. It gets some things removed from it that would kind of just mess up the signal, and then run through machines that can detect what the chemical composition is. Oh, I saw you first. Yes.
How can you tell if the wear pattern is due to the age of the animal or to the more abrasive food that they're eating?
Yes. Okay. So I didn't want, like I said, I didn't want to spend a lot of time on that because the isotope stuff is the bulk of what I've been working on. But essentially the way mesowear studies work is they take multiple individuals from a species and they'll kind of rank based on which teeth are exposed. They'll essentially give each mandible they're working on an age category. So there's younger ones and there's older ones, and you have that whole spectrum. And you would expect there to be more wear on the older individuals, but exactly what age category, when in time that animal starts to wear its teeth out is really what you're looking at. So an animal that's eating a more abrasive diet is going to have much more worn teeth at a much younger age than an animal that's just munching on leaves or fruit. So if you see that wear on a younger animal, that's telling you that it had that more abrasive diet. And you look at, I think we shot for about 20 individuals. We settled for 10 in some cases from each species. You're looking at multiple individuals, multiple species, all from the same time whenever possible, and comparing them across each other. So the younger it is, the more extreme the wear is on younger animals, the faster and more abrasive that diet is. Make sense? Okay. Yes.
Great talk, really interesting research. I'm curious if you've tried using orientation patch count analysis to characterize dental diversity in the assemblage.
I have not.
Okay.
There was a little bit of work done. I don't think specifically with that method, but I do believe there was some like tooth topography type. Yeah, okay. There was a little bit of that done on some of the animals. I can't remember whether it was on the primates or the hyenodons, by another group.
Okay.
And so that is an option. It's just that we're really just getting to a place where we're starting to ask the ecology questions, where up till now, everything's been being described and described. So we're getting there. That's something I want to learn how to do in the future.
So Emily, are you looking at differences in the way the isotope composition changed over time for those sites that have really fast short changes in the Oligocene, Eocene versus those that you had that said they had more gradual changes that even began in some cases before the boundary? So do you see, can you figure out what kind of changes were happening at that boundary by looking at these data as well?
Yes, and we are very reliant on the interpretation of the geology, which honestly is even being argued about at this site. People can't quite agree on these dates, but I'm using these dates for right now because this is the best I have. That's a whole other animal.
Is there at least good agreement on the time span that's out of sight? Like,
Depends on who you ask. Depends on who you ask. But assuming these dates are pretty good, and they're a good guess. They at least give us a sense of where the Eocene Oligocene boundary was, and that we're dealing with probably about 7 million years of time or less. Very, not that long. And what was it, a million or 2 million years, give or take, between each site, depending on where you are.
oh yeah, that's good.
It's relatively short. It's not as short as some sites, but for this time, it's pretty good. And we might shift the dates around a little bit, but it doesn't change it wouldn't change the story that we're seeing of we see the coast recede and then we see the shift into desert forest. It would just shift exactly where that is relative to everything else. So it's always getting better. But as I said, I'm a little bit reliant on the interpretations of geology, which are also improving. Did you have another question? Don't make a lot of questions.
The ecological aspects are really fascinating there, especially the niche shift that you're seeing. Tissue you're seeing. It's great to see more ecological. One of the things I was interested in, you did show some petrified wood.
Yeah.
Were you finding both animals and plants of that?
Yes.
Okay. What types of trees were being found petrified wood?
Some of it's been attributed to mangrove
I see.
wood.
Wow, those were good sized trees.
And there is a whole, right? I know.
Those have to be humongous.
But there is a really good study that shows the pollen, where somebody got into the pollen in the microfossils. And it kind of, it essentially showed us, yes, there was probably angiosperms. I don't remember exactly which was off the top of my head. But there was a forest nearby somewhere. There were mangrove trees around somewhere. It's just kind of a question of what environment those were creating and exactly where it was relative to these localities. The problem is that petrified wood, one of the arguments is whether it's preserved in situ or if it got washed in. Yep, exactly. So yeah, there's a lot of arguments. But again, improving. Yes.
So you mentioned that there was a debate on exactly like what on what like the environment outside of the mangroves have been, whether it was more open or forested. I'm inclined to say more forested because of like mainly what we see in later uh in like the in particularly the Miocene shows like it's just been it shows like a bit of a more rainforest, like not totally, there was still like grassland places, but rainforest still had a pretty big influence. Didn't seem to be until like the later parts of the Miocene where grassland started like taking over.
This is why I kept saying not grass yet, because that is an important thing that happens in the Miocene. And we don't see really grasses in the pollen. You see my seagrass, microfossils of that, that's nice. But you don't have grasses yet. That doesn't stop you from necessarily having an open environment around. And so that really was part of the question, mostly based on having some of the larger animals that were there. But when you don't have a lot of plant fossils, and when you're not really sure what the vertebrates were eating, because you're so far back in time that none of the modern rules apply, it's hard to say what that environment was until we start pulling in methods like this. So it does look like it started as that more coastal, some open environment there with an increasingly dense forest as you go through time. That did happen. That was an important part of the Miocene. And we know a lot more about the Miocene in Africa than we do the Eocene and Oligocene. Yes.
You mentioned the mangrove, they're more of a brackish saltwater plant. So then these animals that would be living in the water there, they would also be, or, using the water, they would also have to be a saltwater type of animal.
Yeah.
So with the hyraxes, I mean, I sort of pictured them being in a more lake or river or something like that. For some reason, I don't know.
I mean, most people I talk to don't even picture them swimming.
Right.
Because modern hyraxes hop around on rocks and hop around on trees.
Yeah.
They don't swim. But they don't have to swim today. That's not something that's really available to them. It's weird, and it's really weird to see them kind of in that very marine looking space. The other possibility is that they just had a really specialized diet wherever they were living, and they were super picky and only wanted to eat one thing, which might be why that one type of hyrax we don't ever see again after that time period. That hyrax goes extinct after that. Other hyraxes do fine, but that one's gone. So that's a possibility. But it's also possible that it was doing just fine swimming around and eating seagrass, and things do live in that environment. Turns out if you're adapted to a really salty environment, you also you have adaptations that work out later in a drier environment. So I have a lot more questions about that one, but there has been work done on them before. I'm not the first one to get data back that's, oh wow, that's really narrow. They look like they could be swimming. We're just really reluctant to say it because these hyraxes don't swim in the modern day.
And the fact that they're related to elephants, you don't see elephants in the oceans or salt water.
You don't see them in oceans or salt water, but they do love swimming.
Oh, I know that.
They absolutely love swimming. And zoos have, like, the one zoo in America, I think it's the San Diego Zoo, that has hyraxes. They've offered them water. They really don't have anything to do with it. Because that was one of my questions as well. Would they swim if given the opportunity? No, they really don't want to be in the water. But. So it's weird, but we're also dealing with 34 million years or 37 million years in the case of Dimonarium. There's a lot of time in there for a lot to change. Yes, sir.
I wanted to add on to could you flip back to the slide that had like all the different hyrax species?
Oh boy, I can try. While I'm doing that, what was the question?
Right there. So there are actually so you mentioned about how moose they don't have the typical aquatic appearance, but they do also like go out to sea. Well, you notice with like stuff like megalohyrax and a lot of them, they seem to have a bit like longer legs. So actually, they are, dare I say, moose-like and moose eyes. And even with that one, I cannot pronounce the name to save my life. I think
Okay, so this is an important thing to mention. These are not all hyraxes from the phylum. These are all hyraxes known in the fossil record.
Of course,
So this guy,
yeah, it's.
sorry, I can't draw. I had to use a picture from somebody else. This guy's not there. But Antilohyrax is one that people have said, wow, his legs look really long. This one isn't biome. Wow, his legs look really long. That guy was running. That was a cursorial animal, probably living in an open habitat, but probably browsing. And that's that one, that's in that time period that was in the more, where things are in that more open space. So that's nice because that kind of backs up. He had room to run, I think kind of horseish. The thing is, these guys were so diverse in the past that they get described as filling every space that modern hoofed mammals fill on the African savanna.
Yeah.
So imagine all of the many types of grazing and browsing that you see there. None of those animals were on the continent yet. They had room to do whatever they wanted. And then as soon as actually in the Miocene, as soon as Africa was reconnected with those other continents and competition was created between hoofed mammals and hyraxes, that's about when the hyraxes started going extinct, especially the big ones. They do not do well in the face of competition, which makes it even more interesting that we're seeing them not do well in the face of proboscideans and infractors showing up. They change what they do in response to who's around them.
But I was just I do know that Kava hyrax, it's obvious. I do know that it's not from Africa. I've learned that it's from like the eastern parts of Europe. But I did want to point out that it did actually have a very tapir-like smic-like niche. So the idea of a hyrax going out to sea with like sort of moose-like proportions and with there being a hyrax that looks like a tapir, I don't think it's that out of question when you really look at it in the grand scheme of things. Just want to throw that out here.
It's not crazy because they were so diverse. There's the space for that. Well, we'll convince people. My advisor always has questions about, okay, you think that was what was happening? Was it feasible in the ecosystem?
Yeah.
So I had to answer that. Anything else?